As our waterways are being increasingly affected by chemical and biological pollutants, estuarine filtration becomes more important. Oysters filter suspended particles such as phytoplankton, sediment, nutrients, chemical contaminants, and bacteriological material out of the water column. Oyster Creek is a small, enclosed body of water that is tidally influenced by the San Sebastian River. It receives stormwater runoff from a drainage ditch that runs through West St. Augustine and has a culvert that helps drain into Oyster Creek. It is also surrounded by 8 storm drains that deposit water directly off of roadways into the water body. The project objective is to identify trends in oyster filtration of Oyster Creek water. The hypotheses that we tested to examine these trends are as follows.
There will be a more significant decrease in chlorophyll A, fecal coliform bacteria, and total suspended solids in the treatment tanks than in the control tanks over time, and that fecal coliform bacteria will correlate with total suspended solids removal by oysters. According to Saylor et al., transport of bacteria via suspended sediments appears to be a significant mechanism operating in the aquatic environment.
To test these hypotheses, we started by setting up 6 20-liter tanks in the lab fitted with identical 80-gallon-per-hour circulation pumps and identical aeration. We set up an LED aquarium light 11 inches above the 6-tank setup on a 12-hour cycle. We collected 7 5-gallon buckets of water from the culvert output at Oyster Creek. We also collected 9 oysters of the same relative size from an oyster bed near the output of the culvert. Once back in the lab, we filled up the 6 tanks with well-mixed Oyster Creek water. We placed 3 oysters in each of the 3 experimental tanks, and the controlled tanks were left with only Oyster Creek water. As soon as the oysters were placed into the experimental tanks, 500 milliliters of water was taken from each tank at time 0. Samples were taken every 6 hours for the 54 hours following, making a total of 10 sampling time points and 60 samples total. Each time a 500 milliliter sample was taken, the water was replaced with 500 milliliters of lab-made salt water. After each sampling, the 6 samples were placed into a refrigerator and filtered within 24 hours. The samples were fitted and analyzed for chlorophyll A, fecal coliform bacteria, and total suspended solids.
For chlorophyll A, 200 milliliters of each sample was filtered through a glass fiber filter. After filtration, each filter was folded and placed into a labeled test tube. The labeled test tubes were placed into a negative 80 degrees Celsius freezer until ready for analysis. Once ready for analysis, the filters were removed from the freezer and 8 milliliters of 90% acetone was pipetted into each test tube. After the addition of 8 milliliters of 90% acetone, each test tube was sonicated to break up the chlorophyll cells. The test tubes were then placed into a normal freezer for 24 hours. After the 24-hour period, the samples were removed from the freezer, brought to room temperature, and the fluorescence was taken using a fluorometer. Final values were then calculated in Excel. For the fecal coliform bacteria analysis, 20 milliliters of sample was filtered through a sterile gridded filter. After filtration, the filter was placed in a sterile petri dish with rosalic acid broth to promote fecal bacteria growth. The petri dishes were sealed and placed in an incubator at 44.5 degrees Celsius for 24 hours. After the 24-hour period, the fecal colonies were counted and recorded. For the total suspended solids analysis, 200 milliliters of sample was filtered through a pre-weighed glass fiber filter and thoroughly rinsed with deionized water. The filters were then transferred to a 110 degrees Celsius oven for 24 hours. The filters were then removed and placed in a desiccator to cool. The final weights of the filters were recorded and the final suspended solids were calculated.
This experiment was repeated with some adjustments to hopefully show better trends in the data. The alterations are as follows. Instead of 3 oysters in each experimental tank, 6 oysters of the same relative size were placed in each experimental tank. Samples were taken every 12 hours instead of every 6 hours, for a total of 72 hours, which led to a total of 7 sampling time points. Due to a salinity of 0 in the collected water, instant ocean aquarium salt was added to each tank initially to increase the salinity to 20 parts per thousand for the health of the oysters. Each tank was spiked with phytoplankton collected at the city marina to increase the initial values of chlorophyll A. At the end of each experiment, the oysters were returned to the collection site.